antibiotics. And when we do, we'll talk about how they work. And then we'll talk about antibiotic resistance. So this will focus on how antibiotics are killing bacteria. Antibiotic resistance is how do those bacteria become resistant to antibiotics. Easy to mix them up. So an antimicrobial means it's effective against a microbe. And a microbe could be a bacteria, it could be a fungus, it could be a parasite, right? There's many things that are microbes. Or microorganism. Now a fungus was found to secrete penicillin on a petri dish, right? Fleming in the 1940s figured out his petri just got contaminated with fungus. It secreted the antibiotic, and then the bacteria wouldn't grow on the plate where that antibiotic was. So that started the antibiotic when he purified it and we started using it. And we noticed that it wasn't the only, that fungus wasn't the only microorganism that produced molecules like penicillin. So we consider penicillin a natural metabolic process. It's normally found in nature. Most of the ones that we use are. And they're way, it's sort of like the fungus way of keeping people from eating their food in the soil. Right? Because a lot of these bacteria are found in soil and fungus too. Before antibiotics, there were antimicrobials like sulfur, which is a chemical, sulfonamide, which is a chemical that interferes with the bacterial metabolism, and I'll explain that a little bit. But doesn't meet the definition of an antibiotic. There's broad spectrum and narrow spectrum. So if you consider an antibiotic broad spectrum, it just means that it's effective against a broad range. And what broad range means, if you looked at, these are 5 different groups. Mycobacterium gram-negative, gram-positive, right? Probably over 90% of the medically relevant or the infections that most of us get fit into these 3 categories. Chlamydia, there's a lot of chlamydia around for sure, right? But it belongs in its own category, and so does rickettsia. Causes Rocky Mountain spotted fever. But tetracyclines work against a broad range in this depiction, whereas narrow range would be like tobramycin, smaller. There's not that many antibiotics on here. But if you think of things like, Pen G or Pen V, they are very similar antibiotics. It might depend on, Pen G might be more effective against urinary tract infections than penicillin V. Or penicillin V might have the V because it's given IV, and penicillin G can't be given IV. So there's all kinds of reasons why you give them different names. Because you need the antibiotic to reach wherever the infection might be. But a target for the antibiotic is where in the bacteria is the antibiotic binding or affecting. So if it affects the cell wall like penicillin, then the bacteria have more trouble making their cell wall. Many times, right, in that case, it's inhibiting enzymes So it binds to the enzyme that you need, that the bacteria needs to make the cell wall, and when it does, it inhibits the activity of that enzyme. Now, a ribosome is used for protein synthesis. It's sort of an enzyme in a sense, right? Because it puts amino acids together. But antibiotics can target the ribosome, and if it does, then it affects protein synthesis of bacteria. Penicillin targets the cell wall by blocking cell wall synthesis. And just a reminder, the cell wall contains sugars, NAM and NAG, that are linked together. And then the little green circles are small peptides, which are just each green circle here represents an amino acid. So you have to put those amino acids together. So there are probably 3 or 4 enzymes all together, and any one of them, right, would be a problem for the bacteria if they didn't work properly. A bacteria with holes in it, not so sure it's that clear cut. So with the antibiotic, you would expect the bacteria just to have trouble making their cell wall, and they might lose the integrity that the cell wall gives them, right? If you take penicillin and you don't have an immune system fighting the infection at the same time, it may not be effective, right? We know that antibiotics alone don't really do the job. They will on a petri dish, but maybe not in your body. So penicillin-like antibiotics, and I think you can, if they have psyllin, I think those are considered penicillin-like. Lactam ring is part of the penicillin molecule. And when we talk about antibiotic resistance, called penicillinase, and it breaks this molecule down. But we'll talk about that then. But be sure you see this is different than penicillin. Then these are 4 antibiotics that target the ribosome, and they have specific spots on the ribosome where they stick to. And when they do, then they block synthesis, protein synthesis. A sulfonamide drug is not an antibiotic, but it is an antimicrobial. Sulfonamide, right, is the general name. And what they do is they inhibit an enzyme for this pathway here. Right, and then folic acid is what's required to make many things in the bacteria. So if you don't make the folic acid here by inhibiting this enzyme with the sulfonamide, then the bacteria has trouble making those DNA, RNA. But we don't think of a sulfa drug, technically it's not an antibiotic. So these are antibiotic mechanisms. Now we want to get into mechanisms of how bacteria become resistant. So we used, I think, penicillin in the early 1950s. And I think within 2 to 3 years, they started noticing that bacteria became resistant to penicillin. And it's because they started making penicillinase. All right, and that, right, is this part right there's the beta-lactam ring again, and it's just showing you part of the penicillin molecule here. There's more to it, but the penicillin breaks this bond, right? And so we say that the beta-lactam ring is broken by penicillinase. That renders the penicillin inactive, right? And so that's one mechanism to get antibiotic resistance. Drug inactivation. Decreased permeability, meaning that they develop a way. A good example of that might even be a capsule. If they secrete a capsule around their surface, then the antibiotic can't get in. They show it. You don't have to understand the principle of what's happening here. You know, they're showing it's using a receptor of some type to get into the bacteria, and then that receptor becomes mutant. And number 3 is they can activate their drug pump. A drug pump is a protein that pumps toxins out of cells. So they would, bacteria considers an antibiotic, a toxin, of course. But if you can inactivate it, or activate it rather, which means it'll pump more of the antibiotic out, that would be another type of resistant mechanism. And then the 4th, changing the binding site, and they use the ribosome target as an example. Right, if this is where, if you mutate this part or change the shape of the ribosome so that it has a little indentation. All right, so if you had a ribosome is supposed to have this shape and the antibiotic binds there, if it altered the shape and now the antibiotic doesn't stick to the ribosome, then that would be another changing the binding site of the antibiotic, right? Or changing the target enough so that it no longer sticks. Now, MRSA is, we're going to talk about that in a second. It stands for methicillin-resistant Staph aureus. This is an enzyme, rather, that the bacteria needs to make the cell wall. So this is a component of the cell wall here, and in order for this enzyme to put the cell wall together, or the components, they have to bind to the active site. Right? It's actually the cross-link, so it's the peptide that it's cross-linking. But if you give the bacteria methicillin, or methicillin-like antibiotics, then they stick to the active site of that enzyme. That would inhibit cell wall synthesis. If they get resistant to the antibiotic, they're still able to make the cell wall with the change, but right, the methicillin no longer sticks to the active site of the enzyme, so it's no longer effective. Right, it can't bind to the active site like it did prior to, right, that would be an altercation of the active site, means that there was a mutation that now keeps, they call it the beta-lactam antibiotic, right, which are, like I said, penicillin-like antibiotics. So they call it methicillin resistant, but it is resistant to other antibiotics. I think you have to use vancomycin. Alternate pathway, that means you want to get around. Now I described the pathway here. A pathway just means, there are several molecules that need to be made to get to the end product. So if you can get around, like if this enzyme that converts PABA to this, dihydrophthoric acid, right, then if you can make PABA with something else and get around it, that would be, an example of an alternate pathway getting around. I listed 5. All I'd expect you to know is to recognize any one of those 5 as a antibiotic resistant mechanism. don't worry too much about, how they work. Now, a way of determining resistance is to, like we showed you before, right? I said, you can put these discs on a plate. Before you do that, you cover the entire surface of the plate with bacteria, and then place discs on the plate that contain various antibiotics. If you're, say, have an infection, the doctor orders a CNS, culture and sensitivity. Culture means, culture up and identify the bacteria. Sensitivity means determine whether the bacteria is resistant to usually it's like 10 different antibiotics. Every lab has their set of antibiotics they test for, right? There's just 6 discs on this plate, so they're only testing for 6. They're labeled to help you see, right? C means there's chloramphenicol. AMP is ampicillin in that one. The green area, they let the bacteria grow, and then the green area up here are where the bacteria was able to grow, right? I showed you the zone of inhibition on that other, in section 10, with the disinfectants. But you would expect the bigger the zone, right, the less resistant. If there's no zone at all, then the antibiotic isn't working. Okay, so the zone of inhibition is generated by the antibiotic in the disc. They put in here, this one is actually considered resistant because the zone of inhibition isn't big enough. Right? But this one is big enough. Intermediate means it's in between. Right? This one, oxytetracycline. Right? The bacteria that grew on this plate is resistant to oxytetracycline. And the gentamicin and the ampicillin are I's, which stands for intermediate. They have it on here? No, it's not. And intermediate just means it's neither resistant or sensitive. It's in between. And then this would be a table. And if we look down here at vancomycin, in order for the bacteria to be susceptible for vancomycin, then the zone has to be larger than 12. So, I don't know if they don't tell you the size of these, do they? That one up there, I think, this one's 27. But the zone would have to be, when you measured it, it'd have to be greater than 12 to be susceptible. If it's smaller than 9, then it's resistant to vancomycin. And if it's in between 12 and 9, it's intermediate. Methicillin-resistant Staph aureus is what MRSA stands for. Now, HA is healthcare associated, right? CA is community associated. So I kind of want you to, I think there's a question about healthcare associated infections, and we'll talk more about it in the next section. The old name for this, and it's still used today, I'm sure, nosocomial. A nosocomial infection means you've gotten the infection while you're at the hospital or some medical facility. And one of the things that any good hospital does is to want to keep nosocomial infections to a minimum. Right? And they would probably put a lot of pressure on nurses to make sure, right, they do, right? Now, if you're liable, right, as a hospital, as a medical facility, if someone were to die from a nosocomial infection, you could really sue the hospital for malpractice, right? Because they got the infection. It's sometimes hard to prove, but And MRSA becomes such a big problem, but if you go to a hospital, they might swab your nose to determine if you have MRSA. And that way they can label you, and then they're taking preventative measures so that they're less liable for, right, if they they get sued, they can say it during the trial, this is what we do to prevent. CA is community associated, and that means, like I was saying, I think nowadays they're probably able now to, because there's so much of it, it's not just a problem because you went to a hospital. Now it's easy to proliferate them. Resistant bacteria contain a gene, or one of their genes become mutated to make the antibiotic ineffective. If you remove antibiotics, then it would probably reverse very quickly. Right? You wouldn't have resistant bacteria. But if you put bacteria on a plate and only 2 of them are resistant to an antibiotic that's also in the plate, right, the resistant ones are able to grow, but the ones that aren't resistant don't grow. Normal flora, if you're given antibiotics, the other problem that you get with it is that it does kill normal flora in your body as well. All right, so in your intestine, you have about 10 or 20 different bacteria growing, and they're all growing at different numbers, right? There's not as many as certain bacteria as other bacteria. Or like even yeast, you might have some yeast in your intestine, but not much. But if you kill out the normal flora, then the ones that are there in very few number can overgrow. They call it a super infection here. So if you find Clostridium difficile, it's a bacteria that makes spores, right? It's usually in your intestine and doesn't cause a problem. But if you're given, normally it's IV antibiotics, and when you get them, sometimes you even get 2 at a time. But they're much more effective than oral. So you don't really see this too much with oral medications, but they get the C. diff. It just means that this bacteria is now producing lots of gas. They're a gas producer. If you're given an antibiotic, then a probiotic are things like yogurts that contain sometimes only one. A couple of them might have 2 different bacteria. Lactobacillus, acidophilus, I think is another one. But they're not the ones that they're normally found in your, right? You need E. coli. You need like 10 different bacteria, right? And you can't eat them. This is, most of the bacteria that are in these preparations are going to die in your stomach. But you do get some of them get past. I mean, it's not like it doesn't help. But if you're taking antibiotics, there's probably a good reason to take a probiotic like this. But if not, now this is another way to get around the problem. A fecal microbiota transplant, right? So microbiota refers to normal flora. And I suppose, if you got a stool sample from yourself, I could see that, but I can't see. I thought this is so funny. MIT has a stool bank where I think they keep it frozen, but you could get a stool transplant. So now, right, there are antifungals. They're good against fungus. Lamisil and amphotericin work. And I would hope at this point you understand you can't use an antibiotic that's effective against bacteria if you have a fungal infection. If you have a helminth infection, right? Flukes, tapeworms, roundworms, and I'm pretty sure this is what ivermectin's used for. It's a anti-helminth or anti-protozoal medication for humans, right? It works really well. It's like a miracle drug for people where these types of parasites kill people or cause river blindness, all kinds of problems, right? If they take ivermectin, not a problem. But you wouldn't take an antibiotic to get rid of a roundworm infection. Right, Giardia or Trichomonas, you would use their protozoans, right? That's a type. They're single cell organisms. And then if you had a viral infection, you would take an antiviral. But an antibiotic would not be effective against the viral infection. So Tamiflu is a good example. Neuraminidase is the enzyme. It's when we did the virus section, I said they have the HA and the NA spike proteins. Well, NA stands for neuraminidase. I think that's how you say it. It's an enzyme, but it helps get the virus into an epithelial cell. If you take Tamiflu, then it interacts or blocks the activity of that enzyme. So you say it works by preventing the flu virus from getting into epithelial cells. That's the mechanism of action of that. There are others, but I'm surprised there's not many more of them. Then there are a class of antivirals that inhibit DNA and RNA synthesis. And the word nucleoside analog, I think I mentioned that before in section 9. They inhibit the enzymes that make RNA or DNA. So acyclovir, ribavirin are examples of nucleoside analogs. And I'm going to mention AZT in a minute. It inhibits DNA synthesis of HIV. Some bacteria require protease. Protease is an enzyme. It digests protein, but not just any protein. It digests one of the viral proteins needed to make the viral particle. So we call them protease inhibitors. They work by inhibition of the viral assembly. So in order to assemble the virus, you have a bunch of proteins that form the nucleocapsid. But before they can, say, stick together like this, you need this protease, and the protein may look like that, and then after this enzyme works, this protease, right, you get just one circle. As long as it's one circle, you can form the viral nucleocapsid, but if it's still together as two parts, then it can't form the viral particle. So a protease inhibitor inhibits the protease, right? This doesn't happen. They stay together, A and B, and then you can't get that occurring. You don't assemble. So HIV requires a protease inhibitor. I don't think coronavirus has a protease. So it's just some viruses that require this type of an enzyme, And if they do, then they classify drugs that inhibit protease inhibitors. So tolapivir and simivir are good for hepatitis C, I think. Harvoni was, I think, they started using that, I think it was like 3 or 4 years ago. Now, I think there's even a better drug than that. And I think when they came out with Harvoni, they were still using interferon. But they were thinking about getting rid of the interferon treatment because many people, they make you sign paperwork that you're going to complete the. Now the treatments for hepatitis C are usually something like Harvoni that have two different, right, it's a cocktail, more than one medication, but without interferon. There was a time when interferon and ribavirin, I think for like 2015 to 2008, you know, for like 3 or 4 years that was used. And then AZT was the original medication for HIV, and unfortunately they killed a lot of people because they had them take way too much of it, right? I think they were taking 5 or 10 times more than what they would be given today. But they started using this in combination with a protease inhibitor, and that's where the term cocktail originated, right? When they started using 2 or 3 different medications to treat HIV back in the 1990s, inhibits the reverse transcriptase, which converts RNA into DNA. And if you inhibit that, then the virus can't replicate. So it works pretty good. And the protease, HIV has its own protease. And like I said, there's inhibitors. If you inhibit the protease, then you block the ability to replicate the virus. That's how those medications